Hey all, welcome to Shatrick. This is Raj here. I thank you for your patience for waiting until 15th of November to see this video. So today I'm going to present my discussions with Jeff Galvin, founder and CEO of American Gene Technologies. It was a really wonderful experience talking to Jeff, and I was able to get a lot of information about AGT 103 and the other therapies, as well as the platform that Jeff is developing in AGT. And I'm pretty sure that you're also going to learn a lot by watching this interview. And I know what you're thinking. Yeah, you would have asked a lot of questions. I think I have covered most of the questions you would have asked if you had the opportunity to meet Jeff. But if there are some other questions that you have which I have not yet covered, please feel free to put it in the comment section. I'm going to gather them all together. And Jeff has been gracious enough to allow me to approach them again somewhere around January of next year. And at that point of time, we could have another interview, and I could put those questions across to Jeff. And also, if you look in the description section, you'll find a link to the newsletter section for AGT. So if any of you are interested in getting up-to-date information from AGT, you can just follow that link and go and register on the website of American Gene Technologies. And you will start getting the newsletters that they have. I think they have a quarterly newsletter. So that will keep you updated. With that, my friends, let me present the interview with Jeff Galvin. Let's get started. Today, I have the pleasure of introducing Jeff Galvin, founder and CEO of American Gene Technologies. Let me provide you with a brief introduction. Jeff earned his BA degree in economics from Harvard in 1981 and started in the computer industry and has had the good fortune of having worked with the great Steve Jobs. Jeff has more than 30 years of business and entrepreneurial experience, not only as a founder, but also in executive positions at a variety of Silicon Valley startups. Several of Jeff's companies have been taken public or taken over by public companies, including one in the medical technology arena that was sold to Varian, the leading maker of linear accelerators used in cancer therapy. Following his startup experience, Jeff retired to become an angel investor in the real estate and high-tech business. And an impactful and chance meeting with Roscoe Brady at NIH inspired Jeff to get back on the saddle, and the result was the founding of American Gene Technologies. Welcome, Jeff. Thanks for giving us your time and joining us today.
My pleasure. Thank you for having me, Raj.
Thanks, Jeff. Can you please talk a little bit about the AGT origin story, a little more color on the meeting with Roscoe Brady at NIH, and what inspired you to start AGT?
Sure. So I retired from Silicon Valley in about 2002, I guess, 2001, 2002. And I bought a house in Hawaii. And I had this, what I would consider kind of a dream retirement life, flying back and forth between Silicon Valley, which is really beautiful, and my house across the street from a gorgeous beach in Maui. And I would say the only problem was that I was only 42 or 41, 42 at the time. So it got boring. And I wanted some stimulation. Just for my brain, and I thought I would just dabble in high tech a little bit more. So my initial thinking was that maybe I would make a small investment in a Silicon Valley startup, or sit on a board, or mentor an entrepreneur, or something like that, part-time work. But when people find out you have money, you get all these incoming business plans, and one of them came from NIH, Roscoe Brady's lab. It wasn't Roscoe Brady who sent it to me, but one of his postdocs.
Right.
And it was kind of intriguing. And I went out there to visit, and this postdoc introduced me to Roscoe Brady, who explained viral vectors. And viral vectors, as you probably know, and maybe some of the people in your audience, you can actually crack open deadly viruses now. You can scoop out their deadly DNA, the things that cause diseases, and throw it away.
Yes.
You end up with an empty shell that can deliver anything, like a stealth bomber, and you can put whatever payload you want in it. So now you can put things that improve your body inside those viral vectors, DNA that can actually be added to your existing DNA to enhance the capabilities of your cells and to fight disease instead of creating disease, essentially converting viruses into updates for your cells. Well, coming from the computer industry, my head practically exploded. I just said, Dr. Brady, you've just shown me the diskette for the organic computer, the human cell.
Yes.
And now that we can go in there and we can update the operating system, the DNA of the human cell, what can't we cure? And I just got so excited. I just couldn't help it. I'm not a biotech guy, or wasn't at the time. Now, 15 years, I consider myself a biotech guy. But back then, all I knew was that I had this incredible sense that I'm at the beginning of a complete revolution in pharmaceuticals, that the power of this, the specificity of it, that we're changing exact little things in your body. We're not just pouring liquids into your body and hoping, that the good effects outweigh the bad effects. No, we have now viruses that can be targeted to specific cells that will make very specific changes in a targeted manner. And these changes are, how can you get more powerful than changing DNA? So, like I said, I just saw this incredible future for gene and cell therapy, and I just couldn't resist it. I took all the money that I had and started the company. Yeah.
That was a big risk to take, but you did. And I think many people are fortunate you did that, because we have AGT going into a very good situation right now with the clinical trials.
Yeah, well, keep your fingers crossed. We're not all the way across the finish line yet, but I would say that, yes, we have tremendous momentum right now, and it's in a couple of very important areas. One is in a potential HIV cure.
Yes.
But you got to remember that this potential HIV cure is actually built on top of a platform of a whole bunch of reusable components that you can mix and match to create the foundation of many cures. Yes. So what's really exciting about HIV is not just that we may be able to provide hope for many people that have been diagnosed with HIV, but if we're successful in that, we'll take that money and reinvest it, and we can cure a whole bunch of other chronic viral infections, hepatitis B, human papillomavirus, HTLV, but also we have assets in that platform in immuno-oncology, and I think we have a high potential to make a big difference in cancer. So, I look at us as being the Microsoft of this new software for the organic computer. And you probably understand that Microsoft ended up being one of the biggest companies on earth, because their platform, right, MS-DOS originally, yes, Windows, whatever, is part of almost 80% of the applications that are out there, right, are running on PCs. And so and why would you use Microsoft instead of writing directly on the machine? Well, that's obvious, right? Microsoft provides all the basic functionality for creating an application, and then you can focus on the creating value on top of that. Yes. Well, we're doing the same thing for gene and cell therapy. We're providing a framework of tools that allow you to accurately deliver therapeutic levels of any gene expression to the right cells in the right quantity safely, while avoiding the healthy tissue. Right. So after that, what you can do is you just decide what needs to be modified in what cells to mitigate the disease. And that's how you create your application without having to reinvent all of the different components that allow you to do that delivery reliably, accurately, and safely. And so I think we've got the MS-DOS for your body right now, but we're going to turn that eventually into the iPhone for your body, where curing any disease is like writing an app on an iPhone, because it does so much of the heavy lifting. So yeah, I'm very excited about it. I think the momentum is very good, but it's still hard work and a ways to go. And but I'm remain optimistic, excited, and engaged. Yeah, many people are very excited about the way things are going so far. And Jeff, could you also please give us an idea of what went behind selecting these therapies that are presently in your pipeline? You have 3 therapies in your pipeline, of course, AGT-103, and you have 2 more, one in oncology. So could you give us a little bit more color on that? Yeah, so when I started the company, I decided that the world was not ready for gene and cell therapy. That included the FDA. They didn't know what to do with it. They had all sorts of special restrictions. There had been some really high-profile accidents, as a matter of fact, where the FDA and the industry and investors had all pulled back. But I believed that what we had seen was the initial overexuberance, like irrational exuberance, followed by failure, followed by irrational sort of pullback. Right, irrational discipline. So I said, okay, it's just like the internet. I've been through lots of tech curves before. The computers were like this, PCs, software was like this, the internet was like this, apps was like this, everything. All technologies follow that curve. So I said, don't worry, it's coming back. But we have to, be careful, because we could spend a ton of money sort of trying to brute force our way through the system, when in fact it would be better to spend it on things that sort of prepare you for where the world is going, so that you can take advantage of it when it gets there. Sort of like the idea of skating to where the puck's going instead of just chasing the puck all the time. So what I decided was this idea of the platform. What we need to do is research many, many different diseases and find out what they have in common. Learn a lot about that level of it, so that we can deliver that level into all of our future developments, and eventually to other people's developments. They could license our platform, or utilize it on a royalty basis or something like that. So we did about 20 different proofs of principle. And the way that I funded it was with my own money, but also the National Institutes of Health were very, very interested in having gene and cell therapy approaches to many different diseases. So I got a ton of SBIR grants in specific disease categories from different institutes.
Yes.
And so they were kind of paying half to 2/3 of the work, and I was paying, half to a third. And, we were starting to collect all these tools and all this knowledge, and it was getting easier and easier and easier to do the work, because of the experience and because of these components that we were accruing. And then I looked at the industry again, and I said, it's getting ready. This was about 2015. So I said, it's time to get serious about getting to the clinic. And we looked at everything in our pipeline, and we picked out those three particular disease areas, because we thought HIV looked like it was really amenable to the regulatory path, because the way we're treating HIV is very similar to CAR T, and CAR T had a lot of momentum.
Right.
So the FDA was starting to feel like they understand how to take T cells out of the body, modify them, and put them back in, and they understand what the safety concerns are,
Yes.
and most important, what they know what it's not, right? So it was getting more efficient. And so HIV looked really good. Phenylketonuria looked good. Just the data coming back from that, and we had a really good collaborator at the University of Pittsburgh Medical Center. And then immuno-oncology, my feeling was that is the really big future of gene and cell therapy, is to wipe out all cancers. I think we can reduce cancers to an infusion or a shot in the arm one day. We can send radiation and chemotherapy the way of bloodletting and leeches. I can tell your audience, I am 100% confident of that. I can't promise we'll be the company that does it, because we have to continue to be successful and maintain this momentum.
Yes.
But I know it's coming.
Yes.
And it's the same feeling I had with computers. I saw the future of computers when I had my first experience with them in 1970. And and the same thing with the internet. You recognize when you're at the beginning of something big, and you understand that the longer you wait, the capabilities of that technology go up and the costs go down.
Right.
And so in that environment, if you can dream it, you can do it.
Yes.
So we took these three disease categories that we thought, with HIV, we thought, okay, that one looks really good for the clinic. Phenylketonuria, we thought the data looks really good on that. And immuno-oncology, we thought this is just too big to ignore, and we had assets there. And those are the ones that we decided to progress. Then, once we were doing that, when people saw the HIV data, they got very excited, and we got all these amazing consultants and advisors and employees, and they started dotting the I's and crossing the T's on that therapy. And that's why it was sort of like blessed in a way. It just got incredible momentum. So it's the first to the clinic, and there we are.
That's really encouraging to see the trajectory of your thought, how you started off from 2015 onwards, and then zeroing on these three therapies. So it's very impressive, and thanks, Jeff, for that. And could you please explain how AGT103T works actually?
Sure. Yeah. So let me back up a little bit to just explain how HIV works, right? So HIV is this really fragile, slow-moving, really weak virus. But what's amazing about it is that it has learned to infect the T cells that are in your body to protect you from HIV. And when it infects those T cells and kills them off, then your body has no way to fight this slow moving, fragile virus, right? That is the only advantage that HIV has over the body. And then we even know that some people have a rare genetic mutation where HIV has a hard time latching onto those T cells, and frequently they're immune to HIV. So they get infected, but it doesn't catch on. Right. They get, HIV in their body, but it doesn't catch on. And that turned out to be because they're missing sort of a door handle that HIV needs to hold on to in order to get in, lift itself into the cells. Right. That's called chemokine receptor number 5. All right. So this sort of started the momentum of the HIV cure industry, because there's a lot of people, and we're standing on the shoulders of some really great experiments and scientists in the past. But what they learned is that if you can get rid of somebody's CCR5, they become highly resistant to most forms of HIV. And so the initial ones were that they did bone marrow transplants. A doctor in Berlin figured out he had a patient that had a incurable leukemia, and the only way to cure them was a bone marrow transplant. So he said, look, if we're going to do a bone marrow transplant, if we take the bone marrow from somebody that's naturally immune to HIV and we do the transplant, we're essentially regrowing that immune person's immune system. The person is immune to HIV, their immune system, in this HIV patient. I wonder what would happen. Well, long story short, that person went into durable remission without antiretrovirals. They were functionally cured. His name was Timothy Ray Brown. I think he passed away a few years ago, but he was able to live almost 20 years without needing antiretrovirals. So that was the first experiment. But the problem with bone marrow transplants is that 6 out of 10 people will die from the trauma of the bone marrow transplant. So it's not a cure for the average person, right? And so you can only get that cure if you're going to die without a bone marrow transplant. Right. And then they still have to find the right bone marrow to transplant you. It's got to be compatible, and it's got to be what's called CCR5 negative. Yes. Right. Okay. So now there's another problem with the bone marrow transplant, is that there are other forms of HIV that don't need CCR5 to get into the cell.
Oh, I didn't know that.
Yeah, a lot of people don't know that about 30% of people that have the most common form of HIV, it's called an R5 strain because of CCR5, it will mutate in their body into what's called an X4 tropic strain, which no longer needs R5 to get into the cell. Oh. Yeah, that's why even when they do these bone marrow transplants and the people survive, not all of them are cured of HIV, because some of them rebound with X4 tropic strain in their body instead. So you need something that's protective against all different forms of HIV if you want to have a reliable, broad cure. Okay, so the next people to give a try on this is a company called Sangamo. Sangamo was kind of a spin out of some work that Carl June was doing at the University of Pittsburgh. And they had figured out a way to pull out T cells, modify them with a virus to strip them of CCR5, and then put them back in the body. And they would pull out all different types of T cells, make that modification and put them back in. And guess what? They were getting about 1 in 10 of them in durable remission. But the problem is they're spending about $500,000 of just viral vector for each patient. Okay, so without any profit, and without all the treatment and the work in actually doing the modification, everything, you figure $500,000 worth of material per patient, and you got to treat 10 patients to get 1 cure. That's never going to sell. True. And they also found that the 1 in 10 patients had to be this genetic mutation called heterozygous delta 32 mutant, which for the layman out there, it just means that you have 2 genes for everything in your body, right? Yes. 2 alleles. Well, it meant that they were already missing 1 CCR5 gene. That's right. What that heterozygous delta 32 mutant is. And then this cell therapy was able to eliminate enough of the other ones that in 1 in 10 cases, these people were cured. So that was not a non-starter. So we took what they're thinking and we added to it. So the first thing that we added is we said, look, we can strip CCR5, but if an X4 tropic strain gets into the cell, how do we prevent it from actually being effective and killing that cell? When we deliver our modification to the cells that we pull out of the body, so it's similar to Sangamo's study. We pull out cells, we modify them ex vivo in a machine. It takes about 11 days, but our virus that modifies them strips the CCR5 and it puts in a short interrupting RNA strands against conserved regions of the HIV genome. So let me translate that for your listeners. What it means is HIV is a series of genes that get installed in the cell that then make HIV. Okay, so what we do is in the cell, we create a negative message from the DNA to those specific genes. And we're knocking out 2 genes in HIV, which make it impossible for it to reproduce itself, no matter what strain of HIV it is. So we strip CCR5, so we have protection, so it doesn't get past the membrane. But if it gets past the membrane, we still are knocking it out before it can integrate. And even if it integrates, those siRNAs will actually prevent it from expressing. So the cell behaves normally. HIV can't essentially co-opt the cell. Because think about it, when HIV gets into the body, it infects the T cells that are the sentinel T cells that are there to protect you.
Yes.
And it converts them into soldiers in the HIV army.
Yes.
They start producing HIV instead of doing their job. Right. What we've done now is we protect those T cells. And those T cells are very, very important. The CD4 positive T cells are what I like to refer to as the conductor of the immune orchestra.
Yes.
It detects the pathogen and it signals to the rest of the immune system what to do. So if we keep the general, if we keep the conductor in the game, right? If we protect the conductor, he keeps the band together. Yes. And what you get is a normal immune reaction to HIV. And remember what I told you? HIV is a very fragile, slow moving virus. Right. So it's not even as hard to clear as a cold or the flu once you can restore normal immune response to HIV. So that's what we did put in our product. And our initial phase 1 shows that the immune system is acting the way that it's supposed to. So we could do this modification. We did it on 7 patients, and none of them had a serious adverse event. So we were able to demonstrate safety. And our secondary endpoints were what are called blood markers of efficacy. We wanted to see that the cells engrafted and that they persisted in the body. So the body didn't eliminate them, and that they didn't get infected. And what we saw is a very, very encouraging response. So as we measure these cells, they come in at levels that are 7 to 30 times normal. So you have a lot of these, conductors. Yeah, HIV specific CD4 cells that cannot be killed in the body. But then they seem to see something that looks like HIV. Now, it may just be impurities left over from the manufacturing process. Right. But in any case, guess what we see next? The CD4 cells start to replicate themselves and increase.
Wow, that's a very positive thing.
Very positive. And then we see the CD8s follow them. The CD8s can go to hundreds of times normal of baseline. Okay, so what we're seeing there between CD4s and CD8s is what? A normal immune response, an effective immune response to HIV. So now the final test of whether this thing works is to take people off their meds and see, take them off of antiretrovirals and see if they can naturally suppress their HIV without those antiretrovirals, right? But I would say, unfortunately, because we didn't file the analytic treatment interruption as part of the phase 1, we did it as an add-on afterwards. And the amount of time between when we did those initial infusions and when we pulled people off of antiretrovirals was on average 280 days. Right. Well, you can imagine when your body has too many of a cell, guess what it does? It eliminates them. So your body looks at it and goes, I got a lot of HIV CD4 positive T cells here, and I got no HIV as far as I can tell. Right. They're not stimulated And why is that? Because you're on antiretrovirals.
Right.
So it eliminates them, and we see a steady decline. At 180 days, it still looks pretty good, but at 280 days is too long. We think the optimum time to take people off is 14 to 28 days. So you can see we're off by a factor of 10 in this study.
Right.
But we still did see some response. So when we pull people off of antiretrovirals, we're seeing that the immune system is responding by increasing CD4 cells and CD8 cells. And we're also seeing in some cases that the normal viral rebound is much lower and takes much longer. So that is very encouraging. And that will be the next thing that we're going to do is we'll treat some more patients, but this time we're going to take the patients off of their antiretrovirals quickly. And what we're hoping is that that will be enough of those T cells to stay on top of the virus so it never rises up to a level where we need to put them back on antiretrovirals. Anyway, that was a long explanation, but the idea is all we're doing is putting body armor on the CD4 positive HIV specific T cells so that HIV can't infect them. If it can't infect the T cell does its job. If the T cell does its job, it can clear HIV. That's the theory. And so far, it looks like it's
and to me, it seems like you have done a home alone kind of setup within the T cell so that even if it managed to get in, then there's the RNA to interrupt any replication. So
yeah, that's right. No, I think the T cells are protected very well. We did an experiment and NIAID actually repeated that experiment. When we were submitting to the FDA, we did a bunch of experiments making blood products. So we would get blood from HIV infected individuals and we would modify it using our process. And then we could test it ex vivo. Until the FDA gives you an approval, you can't test your drug in the human, but you can make the drug and you can test it in Petri dishes.
Right.
So we would go ahead and take the modified blood and put it in a Petri dish and dump tons of HIV into that Petri dish with it.
Right.
And the T cells stayed clear.
Wow.
And they were so effective at keeping themselves clear of HIV that NIAID was attracted to the project. And they actually repeated our experiment. They got the same result on their own patients. We didn't touch this. All we did was give them our viral vector and the protocol. And then they wrote an article with us that's in Molecular Therapy. And I think that was a very big boost to the momentum of the project and also everybody's confidence in this, is that NIAID was able to repeat a very key experiment that showed the potency of this cell product that we were making. It was orders of magnitude better than anything that had ever been seen before. So, and then we had to just keep going through the phase 1, and now NIAID is re-engaged and helping us measure some of the results in the analytic treatment interruption.
That's really good. Actually, I had another question for you, which was going to be talking about the common hurdles that face HIV therapies, but you already explained that in great detail when you explained how AGT works. You gave me the background, gave our audience the background about how HIV functions in the first place, the nature of the virus, and how AGT works. So that kind of covered that. So the next question I have for you, Jeff, is that many of our viewers in the channel are fans of genomic investment, and many are laser focused on HIV therapy, not only as investors, but also as on behalf of patients that they know. So with that in mind, can you please shed some light on the potential FDA approval of AGT-103T? I know you don't want to speculate on this, but whatever you can tell us that you're comfortable with, it will be very welcome by the audience.
So even though we have what I believe is a very successful phase 1, and I think we will go into a phase 2 pending the FDA's acceptance of our data. So we expect to submit that data to the FDA in February. They'll get our full package from the phase 1. And we're hopeful, we don't see anything wrong with the data, but we're hopeful that they'll be satisfied that we met our primary endpoint, which is safety, and our secondary endpoint, which is blood markers of efficacy. At that point, we'll roll into a phase 2, which we believe we could be treating patients in it's probably going to take till 2024 that we'll be treating patients in our phase 2. In that study, we'll do somewhere between 50 and 100 patients. And hopefully, we can arrange that to be a pivotal phase 2, because that could be enough patients. If enough of them show efficacy, the FDA might be willing to grant a limited license to start going out there and commercializing it. There's no guarantees. They're the final adjudicators of safety. So, that's just our aspiration. But if we have to do a phase 3 after that, it will take still a couple more years. But it could be, if everything went in the best way possible, I could imagine that in 2025, we might get a limited license. That would sort of be doing the phase 3 in market in a way, right? If we add, add a year to that to be conservative. So, 2025, 2026, if things go according to plan, that would be a reasonable guess. Now, as a biotech company, of course, we'll be posting a lot of milestones. So if the FDA accepts the phase 1, of course, that's positive. We are spinning out HIV as a separate company. We are starting to make some plans to see if we can take that company public. We don't have any specifics on that yet. But that one might be able to start to gain momentum under its own sort of energy, right? Its own power. And I'm looking and talking to people who might be executives in that company, board members. The IRS has approved the split. That's important because when you split the company, they can tax you on that. And they said, no, they understand that we're not creating any cash there. And so they're going to let us split without paying any taxes. But then we can raise money and take the HIV company public. And so that's something that I'm hoping will happen in the not too distant future.
Right. And the way I understand after your explanation is that before this interview, I was focused on AGT 103. But now, after this discussion with you, I feel that even more important is the platform that you're developing, which is going to create more therapies for more chronic problems. And that is where the true value is. And of course, HIV cure AGT 103 is going to benefit a whole lot of people. But that's not the whole story. The whole story is much bigger than that. And you have been able to explain to us and our audience in a very nice manner. So now that brings me to the question, which I guess most of our audiences who are coming at it from an investor perspective were looking at. You have partially answered that, but maybe you may want to elaborate a little bit. So the question is, how can investors get a slice of action with AGT? You had mentioned the possibility of a potential IPO with the spin-off, which is just going to have AGT 103 in it. Would you be comfortable talking a little bit about that and maybe let our audience know?
Yeah, so I have to be a little bit careful because let me tell you that there are SEC rules.
Yes.
Of a private company cannot speak about investment to anybody that we do not know is what is called accredited. Right. Now, that's not a super high bar, but you have to have like something like a $2 million net worth, not including your primary residence, or you have to have a household income, I think, of $250,000. A lot of people qualify. So let me just say, for full disclosure, this is not a solicitation, or this is not an offering of shares, and nobody should take this as an offering of shares because I don't know who your audience is totally.
Yes.
But accredited investors make up our entire investor pool. We talk to individuals and they've made private investments in our company. And I think that anybody, including not just accredited investors, but actually retail investors, once we go public, can invest in the company. So, keep an eye on us. Let us know. I suppose there's no harm in people letting us know that they want to be informed if we go public.
Right.
And of course we'll keep a list of that. So if we make plans, people know what day, like a newsletter, right? They can get our newsletter would say this stuff anyway.
Right.
So, that would be a way where everybody who's listening to this could anticipate getting into it. If we, become a publicly traded stock, then we'll be conforming to all of the SEC reporting requirements that will protect, sort of your average investors,
Right.
and they would all be welcomed in at that point. So yeah, that's the story. I hope that's I know it's a bit of a fuzzy answer, but I'm trying to be elliptical about it because I just want to be careful to not, stretch the SEC rules,
Yes.
in a bad
No,
way.
I understand, Jeff. Thanks for giving us so much of information about this, because naturally, if I finish this interview and if I did not ask that question, I would have so many of my subscribers just sending comments and emails saying that, why didn't you ask Jeff when the IPO is coming? So I thought that it was better to ask the question. But just before,
Yeah, for sure.
before I let you go, I just have a question that comes to my mind based on the other genomic companies that I have been looking at. So we had initially been looking at CAR-T for cancer. And then I came across this concept of bispecifics, where instead of having a T cell, it's an autologous approach. It's a molecule which on one end recruits a T cell, on the other end, it latches onto the specified antigen and does the cell destruction. So one of the thoughts I had in my mind was to ask you whether you could consider any of your therapies would go the bispecific route. But after hearing how AGT-103 works, I don't think bispecific is the way for AGT-103. But your other two therapies, would you probably consider that kind of approach? Is it amenable? Because it would be allogenic, more easy to market, and much better.
Yeah, so actually a lot of our technology in the immuno-oncology area is allogeneic anyway.
Right.
Okay, because it's based around gamma delta T cells and NK cells.
Right.
So these could be off-the-shelf products.
Yes.
But okay, that being said, I think it's very clever. You said two things now that I think are very clever, and I'm wondering whether you have whether you have an extensive biotech background that I wasn't aware of. But the one that you just said is, yes, I think bispecifics have a lot of promise in the immuno-oncology area, because what we're trying to do, even with the gamma delta T cells and NK cells, is to get them more activated against the tumor. And the way that our approach is to light up the tumor in a way that makes it sort of infinitely attractive to those cells, right? And so that, we've got some data that says that that works. And some of the challenges there is getting our modification into all the cancer cells, because the cancer cells that it gets into, the gamma delta T cells and the NK cells are killing off very reliably without killing other surrounding healthy tissue. But if we can't get to 100% of the cancer, we're not going to be able to do it on our own. So the idea of dragging T cells to the cancer with bispecifics makes a lot of sense. And also think about our viral particle, right? We're having trouble getting the viral particle into all of those different cancer cells within a tumor. Then we're looking at a whole bunch of technologies to increase that penetration. Because if from what we're seeing in our current experiments, if we can get to 100% of the cancer cells in the tumor, we should be able to eliminate it 100%, right, without surgery, without radiation, without chemotherapy. That would be a big step towards my dream of sending, the chemotherapy and radiation the way of bloodletting and leeches, right? And so, I think that bispecifics are one route. Another route are oncolytic viruses. There's other ways to sort of prepare the cancer in a way that will increase the expression of different types of receptors that our virus can bind to. So increasing the binding that way. The same thing's true about PKU. The big challenge in phenylketonuria, PKU, is that it looks like we're not getting a big enough dose to the liver. So we're seeing that what we're changing in the liver is working against the disease. The disease in phenylketonuria is a buildup of phenylalanine that can't be processed because you're missing phenylalanine hydroxylase. So we're restoring phenylalanine hydroxylase, but it looks like not enough. And so, bispecifics might be useful there too. So we're looking at a lot of stuff. There's some other things in that area as well. But, all of these technologies are good. You really see a lot of new things on the cutting edge, and they're all tools in the toolbox. And cancer is really a team sport, right? Like some combination of these technologies will be in the final solution that, removes terminal cancer as a consideration to humanity. I don't think we're far away from that. I sort of feel like it's in the 1 to 2 decade range where all those terminal cancers are eliminated.
Okay, Jeff, before you go, what was the second clever thing that I said that you are referring to?
So you mentioned that you realized during this interview that the platform was really the bigger contribution to medical science. And that's exactly what I believe. And I think that, when I started this company and I was talking about gene and cell therapy like it was software for, the DNA in your organic computer, the human cell, people kind of brushed that off. People that were really experienced in drugs and in biotech said, people don't buy platforms, they buy drugs. Throw away the idea of a platform, Jeff, pick a drug and get into the clinic. But that would sort of be like Microsoft saying, okay, forget about MS-DOS. Let's just write Windows right on the hardware. No, this platform creates an efficiency that is already reducing our costs of doing gene and cell therapy by a factor of 10 above what other companies have. And so, and you look at gene and cell therapy, it's even cheaper than a normal drug development. So you could see a day with our platform that the gene and cell therapy industry could become a garage startup industry where people get, a PCR machine and a cell culture hood and you need maybe something cleaner than an ordinary garage, but you can kind of start these things.
But I get the idea.
Yeah. And anybody with knowledge of a specific disease would be able to take our platform and add their knowledge of that specific disease and create a disease curing application. That to me is the future of pharmaceuticals in the gene and cell therapy space. And I think that's a place where AGT is really in the lead. We have a vast IP portfolio of these components. And I got to give you kudos for recognizing that. One of the reasons we're splitting off HIV and taking that public is so AGT can stay private and nimble and move really quickly to continue to develop our platform from MS-DOS to Windows 11 or an iPhone of disease apps. And I think that is the thing that I'm most passionately excited about, because I think we can not only help potentially 38 million people who have been diagnosed with HIV, but that might just be the beginning.
Yes, that's the tip of the iceberg for you. Because I'm also from a technology background. I used to be a project manager for software. So I understand the concept of software development kits. Like Java is a layer which is making machine agnostic. So probably the human body with all the DNA and the amino acid nucleotides, that's the operating system. On top of it, you have the AGT platform, which is like Java virtual machine. And then you can code and you can make components which can be reused. And drug development and delivery will become much faster. The only thing that can slow it down is the speed at which it progresses through the FDA approval process. But your drug discovery, combination, and other things will come from a library of components that can be put together. And since the components are already tested and found safe, the amount of setbacks in any of the therapy development will be reduced to a great extent, and success factor becomes much higher. That's how I look at it.
Yeah, no, you hit the nail on the head. And I like, since I come from the software industry, I understand your Java reference. And I think that's a very good understanding of exactly what we're doing here. But think about this, is once the FDA has seen a component and they know the component's safe, guess what? You build it all up and the FDA is like, well, we know how everything works in there except for this one little piece. So here's what we need to see
Yes.
to feel safe about that one little piece. Well, what's the difference in that? Well, when you do a traditional drug, like a chemotherapeutic, it's a chemical. It's not software like ACTG. You're not programming genes. No, you have a chemical, and the chemical either works or it doesn't work, and it is unique. And so you need to do every single possible test about how the liver handles it, how genetics affect, the uptake of the drug, how other drugs interact with that drug. All that stuff is a standard set of tests. That is why a new drug costs $1 billion to make. We are already reducing those costs to somewhere around 50 million. Think about that. The difference between $1 billion and $50 million. 50 million is a manageable risk, especially when you think that you could have a proof of concept within $10 million. You could write your IND and get it accepted within $2 million. Because you build it on this platform, and so you can move much more quickly through all of the different milestones and to satisfy the FDA that you've got something that doesn't have too many new things in it, right? And they can go ahead and say, all right, we'll approve the IND. Well, if you have an approved IND on a level of money that you could get in an SBIR grant from NIH, right? So you're a professor or a doctor or a scientist that's been studying a disease for 20 years, you know exactly what you need to change in the body. You come to us, we go, yeah, we have the delivery components you need to make that exact change. Okay, you can collaborate with us, and within $250,000, you can have your mouse experiments done. If you don't see a problem in the mouse experiments, you can write the IND for another, couple of $1 million, and do all of the different experiments that you need to do an IND. And then if they approve the IND, you need to raise about $10 million to do the phase 1. Now, phase 1 in gene and cell therapy will give you not just safety, but some level of efficacy as well, because gene and cell therapy is always, the phase 1 is always done in patients that have the disease. So you get safety, but you also get efficacy, which means that there's no phase 1 really. It's like you're going straight into a small phase 2, right? And so that is an acceleration of that whole process too. Absolutely. So $10 million later, you might have first human efficacy. And I would say that most decent entrepreneurs can raise $10 million.
Yes.
And gene and cell therapy is going to have a success rate of it'll start off 1 in 5, right? No gene and cell therapy really fails because you could keep debugging it and debugging it and eventually get it.
You always develop some intermediate IP which has got value. Somebody can take it and run with it. So
that's
right. It's always there for the 1 in 5 companies.
So there's never real failure, just like there's never been a real failure of a computer company. It gets swallowed up by another one, or a software company gets swallowed up by another one, right? Yeah. So this is the industry that we're in. And so, right now, initial companies that can go through that process and raise $10 million, let's say 1 in 5 of them will actually get a drug.
Right.
So they'll turn $10 million into billions. That's a great, with only an 80% risk factor, that's investable, right? And then it'll become 2 out of 5, and then it'll become 2 out of 3. And pretty soon we'll be able to simulate your whole body in silico. And you understand what I mean by that, right?
Yeah.
So that 99% of the drugs that go into a phase 1 will end up being approved eventually. Right? And the risk will come down to almost zero, and the industry will explode with cures. And there are 10,000 diseases and only about 500 cures out there today. I think gene and cell therapy may cover the rest of them.
True.
Yeah.
This is very, very encouraging. And I'm sure that most of the viewers who are watching this can see the entrepreneur's mind at work. It's people like you, Jeff, who make a difference to this world because you take the risk, you have put your own money, you have followed your vision, and you have assembled a team of like-minded people, good caliber people. And now you're taking it to the next level. And with every bit of your success, you're adding to humanity. So it's a life well led, and it's a life well invested for the good of everybody. Thank you so much for doing this, Jeff.
Oh, yeah. It's a very fulfilling job, right? If I could afford to work for free, I would. I mean, because this is really, think about how exciting it is to be in a position, And I think everybody in my company feels this way. And I think every investor feels this way, and every board member and advisor and scientist that works with us, that it looks like we have a really good opportunity to make a positive difference in many people's lives. And that is just personally fulfilling. It's motivating. It's a reason to jump out of bed in the morning and get down to work and to have fun doing it. So yeah, I'm very pleased to be on this journey, and I really appreciate that you know, you see, you know, some of the positive things in it.
It comes out from you when you start speaking, Jeff. It's very inspirational, and I'm sure this is the greatest asset for AGT, because your team, whenever they are going through challenges, if they just need to speak to you and they get pumped up again, and they'll go back and they will be able to do what needs to be done. So
I hope so. I really believe in this amazing future, and I'm very lucky to have an amazing team behind me. And you know, I think we have a very high chance of getting there.
And before I let you go, Jeff, just one quick question to you. Since AGT is a private company, right? I usually follow public companies which are quoted in the stock exchange. They have their quarterly release when they talk about their clinical trials and milestones and everything. We won't have that visibility. So is it possible that at a later date we can meet again and get some update either through you or your team members or somebody?
Oh, absolutely. I'd love to have another talk with you in the future. Sure. When we have some more news. And I would say, you know, around January timeframe, we'll know all the data from the ATI. So I'll be able to give more detail on, you know, how people did without their antiretrovirals. The other thing I would say, if you don't mind me mentioning this on the broadcast here, I would love it for anybody to go to our website and sign up for our newsletters.
Yes.
And so it's americangene.com. And then just find, you know, the newsletter sign up. And you can find newsletters in the media section, I think, or resources. And then, you know, you'll be getting usually quarterly updates, and we do update people on everything that we can release publicly.
Yes.
And then the other thing is we do a lot of posts on social media. So if this is the sort of thing which, you know, people want to hear things about it, you know, daily or, you know, every once in a while throughout the week, just sign up for, you know, either my LinkedIn or my Twitter or the company's Facebook or LinkedIn or Twitter or whatever. And you can dial it up to whatever level is interesting to you, and we'll keep you informed.
What I'm going to do is I'll reach out to Barry and get a link for your newsletter and put it in the description of this video, so that everyone who's interested can go and have a look at it. And that should be very helpful. And of course, thanks for letting us know that we can get in touch with you again for another interview with some more exciting news and developments, maybe in January.
Of course, it's been a pleasure, and I can't wait to do it again.
Thank you, Jeff.
Thank you.
Well, guys, I hope you enjoyed the interview as much as I did, and I hope you got a lot of information. And that's all for now. Please do not forget to subscribe and press the notify button, because I'll have more such interesting videos coming up very soon. Take care. Have a great day. Bye for now.